Okay, so this is the last part of chapter 4. We were talking about the cell and the cell parts, and in the eukaryotic cells we were breaking it down by processes. So we looked at manufacturing, breakdown, energy processing, and then the last one was structural support, movement, and communication. And so this involves a lot of proteins and a lot of cell membrane and cell wall materials. So we're going to start with some of the proteins that are involved. They are grouped together into something called the cytoskeleton because they really give shape and structure to the cells. There are 3 different kinds: microtubules, microfilaments, and intermediate filaments. All of them are involved with some sort of movement. All right, so each one does something unique and something different, so make sure you pay attention. The way these cytoskeletal proteins actually are capable of helping things move is that they interact with other proteins called motor proteins. And motor proteins are actually what are moving, and they're using the cytoskeletal proteins as a kind of like a monorail. So kind of like the cytoskeleton is the train track, and the motor protein is the train moving along it. And so the cell might need to move a vesicle. Remember, we just talked about vesicles moving from the Golgi to the plasma membrane. They would travel connected to a motor protein that then is walking along a cytoskeletal protein to get to its final destination. Evidence suggests that cytoskeleton proteins really help regulate biochemical activities inside the cell. So microtubules are the first one we're going to talk about. They are a hollow rod of proteins called tubulin. So those little yellow dots that you see at the bottom of the page are called tubulin, and they make a hollow rod, okay? And the diameter here can be about 25 nanometers, but the length of these can be very long. They can be from somewhere 200 nanometers to even up to 25 micrometers long. So they really vary in length. They're dynamic. They can grow and shrink as needed in the cell. Microtubules do a variety of things. They help to give the cell shape. They help with vesicle movement, like we saw in the last slide. They also help with chromosome movement when the cell is dividing. And they also are what allow a cilia or flagella to move. So let's go through each of these. We're going to start with the vesicle movement. So when a cell is not dividing, the microtubules are arranged, as you can see in this bottom picture down here, where you have basically microtubules growing kind of out of a center space. This is labeled here as the centrosome. I often call it the microtubule organizing center. Think of it like a train depot, and all of the train tracks radiate out of this train depot. All right, so vesicles that are being made on the ER or the Golgi can jump on any of these train tracks via their motor protein and get to different places in the cell. So if the vesicle needs to be delivered to this plasma membrane down here, it can travel along that cell. Whereas if it travels to the upper part of this cell, that it will get on a different track and travel. Again, the microtubules are basically a train track along which the motor proteins that are hooked to the vesicles are traveling. All right, and they go in whatever direction the microtubule is going. Obviously, this motor protein to move requires energy, so that energy has to come from cellular respiration. So that means if we're in a eukaryotic cell, which we are, that a mitochondria has to be involved in making this come together. So these are all related, interconnected processes. So this is just a cool picture. You can see the microtubule running along here. And then this is the big vesicle that's kind of trapped on top of the motor protein. You don't really see the motor protein, but it's connecting it right through here. Now, this is what microtubules look like when the cell is not actively dividing. So again, when it's not dividing, you have that microtubule organizing center, and then motor proteins in their vesicles can just pick which highway they want to go on in order to get to their final destination. However, when a cell moves from being out of a non-dividing state into a dividing state, the arrangement of the microtubules drastically changes. It actually doubles, so we end up having 2 microtubule organizing centers instead of 1. The microtubules still grow out of them, but instead of growing out towards the edge of the cell to carry vesicles, they grow towards the middle of the cell and they connect to chromosomes. So this is a chromosome right here, and so the microtubules connect to the chromosomes, and then when the cell actually is going to divide, it separates those 2 copies of the chromosome. So this copy will move towards this side, and this copy will move towards this side. So the microtubules grab onto it and then pull it to their respective pole. We'll talk more about that when we actually get to mitosis. Another thing that microtubules do is they power a cell's movement by powering either the long tail called a flagella or the shorter hair-like structures called cilia. All right, so both of these allow the cell to move. They're called locomotor appendages. All right, so how does this really work? Well, the microtubules are long and they're aligned next to each other in kind of a very predictable arrangement. So you see that they're kind of in these long doublets. You can see the doublet here going down, another doublet going down. And then each set of doublet is connected by motor proteins, okay? So if they're connected by motor proteins like this, what happens is when it's time to move, the motor proteins walk along one of the strands, are connected permanently to the other microtubule, and that causes a bending movement. All right, so that causes a bending movement, and then that's what causes the flagella to flip back and forth, or the cilia to flip back and forth. One thing to keep in mind, and this is really, really important, there are a lot of little appendages that come off of cells. You got to keep them straight. So far now we've learned about 4. We learned about pili, which are exclusively part of bacteria cells, and their function is attachment. We talked about how it can be a sex pilus and they can share DNA through it. We talked about flagella. There's flagella in bacteria that help a cell swim. There's flagella in eukaryotes that help a cell swim. Functionally, they do the same thing. They help the cell swim. Structurally, they're different. So the flagella in a bacteria is not made of microtubules. It's made of a protein called flagellin. And here it's made of microtubules. So even though they serve the same purpose, they're structured differently in the 2 cells. We also learned about microvilli, right? They are the ones that help the cell increase its surface area, and we'll learn how those are formed next. And then we just learned about cilia, which are also short, kind of look like microvilli. However, their job is not to increase surface area. Their job is for movement, and they're made of microtubules. Make a table, keep them straight. All right. Our next cytoskeletal protein are the actin filaments, or sometimes called microfilaments. Those words are used interchangeably, so make sure you know both. The protein that makes up the microfilaments is called actin, which is why they kind of go back and forth. Now, actin filaments are critical for a cell maintaining its shape. You have actin microfilaments basically fortifying the cell membrane and giving the cell membrane fortification. So you can see how it's very much layers of them right inside the cell membrane, keeping the cell in the shape that it has. In fact, its microfilaments filaments that actually support the structure of microvilli. So they grow up into these fingers, holding them up to increase that surface area. Let me pause. Cilia are powered by microtubules and motor proteins. Microvilli are fortified by microfilaments and are for surface area increasing. Make sure you know the difference. All right, now actin microfilaments also work with motor proteins when muscle contraction occurs and when a cell is about to separate and make two new cells during mitosis. This is just a cool micrograph showing us those microvilli and the microfilaments that are giving that structure. So you can see that it's just an integral mixture of proteins making this very complex pattern. Actin microfilaments are also abundantly found in muscle cells. They actually are like filling the muscle cell, and they arrange themselves in these parallel filaments. It's orange here in this picture, and they are parallel and they're associated with another parallel protein structure called myosin, and myosin has these little motor protein heads, and the heads connect to the actin, and then they bend in their shape, and as they do, they move the actin. The actin doesn't actually shrink, they just pull it, and then that causes a muscle contraction. Actin microfilaments are also responsible for cell movement. So you might have heard of an amoeba or amoeboid movement, where a cell grows these little, they're called pseudopod, fake feet, and they grow them by microfilaments growing in length and pushing on the plasma membrane to grow these little feet out, and the cell kind of blobs up behind it. Microfilaments are also responsible for what's called cytoplasmic streaming. So sometimes you see that there's movement of the cytoplasm in a very circular fashion. So the actin associates with myosin in plant cells, different than animal cells, and causes this cytoplasmic streaming. Our last cytoskeletal protein are the intermediate filaments that serve two purposes. One, they help keep the nucleus where it's supposed to be inside the cytoplasm, and they contribute to junctions. And so what you can see here, this is our nucleus, which would be floating around crazily if we didn't have it anchored. So the intermediate filaments tether it in its place so that it can kind of move, but it doesn't move drastically and float around and kind of bump into things. Additionally, intermediate filaments are associated with junctions between cells to keep those junctions fortified. We're going to talk about junctions in just a minute. Now, we have a bunch of proteins on the inside of the cell making up the cytoskeleton, but we also have structures outside the cell that help with structure and function, connecting and coordinating and communicating. So these structures are the cell walls, and we're specifically going to look at plants, but other cells have cell walls too. The extracellular matrix in animal cells, and then particularly junctions in animal cells. So we know already, we've learned about the cell wall when we learned about bacteria, that a cell wall is an extracellular structure. That just means it's outside the plasma membrane. In terms of plant cells, it's a very good way to distinguish a plant cell from an animal cell, because no animal cells have a cell wall. So what are plant cell walls made up of? They're made up of cellulose. Now think about it for a second. What other kinds of cells have cell walls? Bacteria have a cell wall made of peptidoglycan, and fungus has a cell wall made up of chitin. And chitin and cellulose, remember, are both polysaccharides. So what do these cell walls do? They protect the cell, they help the cell maintain shape, and they also protect it against uptake of too much water. What's interesting about these plasmodesmata, these tunnels through the cell wall, is that they are lined with the plasma membrane. So they're not protein-based. They're actually lined through the plasma membrane. So this is a really interesting structure that has to form. It forms during cell division, and you're going to get an opportunity to investigate that further on your next set of discussion questions. All right, the extracellular matrix is the outside of animal cells. Now, animal cells are not so tightly packed together like we see in plant cells, so there's space in between a lot of animal cells. So it's not that nothing fills that space. Lots of times, in order to communicate and properly connect and things like that, there are proteins that are made inside the cell, squirted outside the cell, and then used to fortify this extracellular matrix. So some of the proteins you see there are collagen, for example. So when you get a collagen injection, that what you're getting is collagen squirted into your between the cells, right, to give it more structure. So if you look at this image here, let me get my pen back on, right, we see the plasma membrane, right, here's our plasma membrane. Inside the cell, which is down here, we see microfilaments fortifying the inside of the cell. We know now that we can have proteins embedded in that cell membrane. And now we're learning that we have proteins outside of the cell. Well, when the cell is making these proteins on the outside, it doesn't want to make it and then let it go forever. So these membrane proteins' job is to hold on the inside to the microfilaments and hold on the outside to the extracellular proteins so that they kind of stay together and don't float away from each other. The extracellular matrix functions to support the cell, cause the cells to kind of stick to each other. It can help with movement of things from one cell to the other and for regulation. In addition to extracellular matrix, we have to talk about how animal cells do connect to each other, because sometimes they have to. So there are 3 different intercellular junctions in animal cells that I want to talk about. For comparison's sake, remember what we just talked about with the plasmodesmata as ways to communicate from one plant cell to another plant cell. And we're going to compare that in a second. In animals, there are 3 different kinds of cell junctions. The first one is called a tight junction. Tight junctions serve the purpose of preventing leakage. So they are very tightly grouped together right here. It's like sewing a hem. And now these particles that would potentially be able to slip through cannot, and the only way they can go is through the cell where they get processed on their way through. If these 2 cells were not connected, then these particles would just get through without anything. So we don't always want that to happen. That's a tight junction. Another kind of junction is a desmosome. All right, a desmosome is a, let me erase that, is like a snap, where a tight junction is more like a zipper. So it only forms a connection at certain places along the cell, but desmosomes are the ones that are connected to the inside of the cell via the microfilaments. And so you can see here there's this kind of protein bar right here on the inside of the membrane, has parts of it go through the membrane, and then it goes through the neighboring membrane. So it kind of snaps the 2 cells together, almost like Velcro. The last are called gap junctions. Gap junctions are very much like plasmodesmata in that they allow cytoplasm to be shared from one cell to the next. Different, however, gap junctions are protein-based. So they are a protein pore, similar to what we saw with the nuclear pore, that aligns on 2 different cell types so that they can exchange material with each other. All right, so let's look at, this is kind of what they look like. They have all these proteins and then a channel that's going to connect from the inside of this cell to the inside of this cell. All right, so just a review, tight junctions are like a zipper. They really, really hem this up closed. They prevent leakage. Desmosomes sometimes they're called adhering junctions. They're more like a snap. They're involved with microfilaments. Oh, I'm sorry, I lied. They're involved with intermediate filaments to keep cells from floating away from each other. Gap junctions are just a way for one cell to communicate with a neighboring cell in terms of sharing cytoplasm or ions or small molecules. Okay, so now I want to show you a video that was made by a bunch of graduate students at Harvard. And what they did was they went through years and years of literature, people who published about cell function, cell structures, protein function, processes that happen in the cell, and basically took all of the information from hundreds of different studies and animated what those groups suggested they thought things were looking like on the inside of the cell. So this is really cool, so enjoy. This is outside of the cell. This is the extracellular matrix. Those are phospholipids floating around. They're still outside between cells. Now we're going to go inside. We go in the cell. Now we're looking at the cytoskeleton. Some of this is actin, some of this is intermediate filaments. Those are actin filaments growing. Here's an actin filament that's getting broken apart by an enzyme. That's a microtubule, like the train tracks growing. There's a microtubule coming apart. Here's a motor protein walking along a microtubule, carrying a vesicle on its back. You can kind of see all the vesicles moving in the background. That middle part back there, that's the microtubule organizing center, where all of, it's like the train tracks coming. These are nuclear pores. These are RNAs coming out of the nuclear pores, finding a ribosome. Here comes the ribosome docking on it, and it's going to start, there it docks. Now it's going to start making a protein. So this is a cytoplasmic protein because it stayed in the cytoplasm. That protein was made and it's now going to go maybe to the mitochondria to function. Now this ribosome is docking on the ER, squirting its protein in the endoplasmic reticulum. So this must be a secretory protein that would likely be packaged into a vesicle like this one and carried with a motor protein along a microtubule. This is our Golgi. You can see incoming vesicles. You can see outgoing vesicles. Now the vesicle got to the plasma membrane, spewed its stuff to the outside, put proteins in the membrane. Now we're back in the extracellular matrix. These proteins are going to communicate with their neighboring cells. Now this is a white blood cell that has to drastically change its shape to get out of the blood vessel, so it flattens like a pancake. That's all cytoskeletal based. Squeezes in between the cells that line the blood vessel so that it can get to the site of the wound. Super cool. All right, email me if you have questions.